Hello everyone and welcome back to my YouTube channel, and in today's series we are going to continue with the part 5, which will encompass microbiology and biotechnology, and this will be covering the simplest route to crack GPAT series part 5. So in microbiology and biotech, you basically have few things that you need to prioritize. The number one is vaccines. You need to know the types of vaccines, whether live, attenuated, killed, is it a toxoid, and you need to know the examples: subconjugate vaccine, polysaccharide vaccine, because nowadays there are so many vaccines: pentavalent vaccine, trivalent vaccine. So you need to know the examples of each and every kind, because it is compulsion that a question has to pop up from the vaccines, so you cannot leave vaccines. Next up comes the immunology. So from immunology, you need to know the difference between the 5 types of immunoglobulin: type G, A, E, D, and M. Gamed, you can name it as gamed. And the structure of antibody: it is a Y shaped, it has two chains, light and heavy, joined by disulfide bonds. So you need to know the different antibody structure and the different terminologies like opsonization, precipitation, sensitization. What are the difference between these terminologies? Complement fixation. And on these principles are diagnostic test work. So Vidal test is based upon agglutination, brucellosis test is based upon opsonization, complement fixation is a test that will encompass the test for syphilis. Sterilization. Next up, sterilization is basically there are a method in which there is removal of the spores of the microorganisms. So there are different types of sterilization: physical, chemical, biological. In physical, ज़्यादा more important is dry heat and moist heat. Along with that, radiation and filtration are also to be studied. The examples: the temperature and the pressure. In autoclave, the pressure and temperature is very important. Along with that, you need to know the indicators: physical, chemical, biological indicators, because those indicators are going to help you to identify. Suppose in the question, in the exam question is directly appearing as Bacillus stereothermophilus is a dash indicator, so you need to know that it is an indicator for moist heat sterilization. Similarly, Royce sachet is an indicator for dash, so you need to know that it is an indicator for gaseous sterilization. So that is important. The next one is diagnostic agents. So there are certain diagnostic agents like Evans blue, for Evans blue, CR51, that are used for plasma volume, total body water. You have deuterium oxide. You have so many different types of diagnostic agents. So you need to know the examples of diagnostic agents and tests. Tests like Vidal for typhoid. You have tuberculosis Mantox test, which is tuberculin skin test. You have Scahan's test. You have Rosewater test for rheumatoid arthritis, Rose Valor test. So this is important. Like smallpox के लिए you have different tests. Austerlichoni test. So there are different types of tests available. Then you need to do the list of antibiotics and bacteria used for microbiological assay. So many antibiotics are derived from various microorganisms like Streptomyces venezuelae, Erythraeus griseus, Penicillium notatum, Penicillium grisophyllum. So you need to know that the sources of from which the bacteria are procured and the bacteria that are used for the assay. Like for chloramphenicol, we use E. coli. For Bordetella bronchoseptica is a bacteria that is used for polymyxins. There are certain bacteria like Micrococcus luteus that can be used for erythromycin. So you need to know the example of the bacteria that is used in the assay. Now assay can be either cylinder plate or turbidimetric or tube plate. So that is important. Next up, you cannot leave is staining. Staining may usually IMVIC test, indole, methyl red, Voges Proskauer and citrate. Then you have acid fast staining, and then you have gram staining. You need to know the basic principles like the primary stain, the decolorizer, the mordant, the secondary stain, and the principle like in gram staining. So there is more amount of teichoic acid in gram positive. There is less amount of lipid content. There is more amount of peptidoglycan, which is a thicker one. So that is why gram positive is purple. Similarly, for acid-fast bacteria, it is specific for mycobacterial species. INVIC tests are positive for enterobacterial species. Microscopy pe rarely questions are asked, but sometimes on definitions of resolution, magnification power, the formula, like the cedar wood oil can give 100x in the oil immersion lens magnification. And the type, like phase contrast, dark field, are they used for unstained or stained? Are they used for living or dead? So that is majorly asked. Next up you have is culture media. So your culture media is basically consisting upon your Different agar, which is a non-nutritive, it is only solidifying. You have different techniques like streak plate, pour plate, spread plate. What are the different types of media? Selective, differential. McConkey's agar is for anaerobic bacteria. You have Wilson and Blair Port media. You have various media that are used for various other cultures and microorganisms, either aerobic, anaerobic, facultative, obligative. So you need to know that. Next up you have is the biotech, core biotech, which will encompass the hybridoma technology. So the hybridoma technology is basically a technology in which we fuse the cancerous cells along with the immune cells, that the B cells. So your cancerous cells are fused with the B cells, so that the antibodies will be produced at a quicker rate. That is why we term it as monoclonal antibody. And this pathway is called as the salvage pathway. So this pathway is very important, and the HGPRT enzyme, hypoxanthine guanine phosphoribosyl transferase, and the HAT medium, which is hypoxanthine aminopterin thymidine medium. So this medium is also very important if you go to see. Next up you have is fermentation. Fermentation is basically not that important, but sometimes questions are asked upon trophophase, EDO-phase, upstreaming and downstreaming process. So you need to know that downstreaming is basically recovery of product, while upstream is basically purification and other types of processes. Tropophase is for primary metabolites. IDO phase is for secondary metabolites. You just need to know the basics. That's it. Fermentation may no need to go submerged. How does it work? Or XYZ type of fermentation. How does it work? No, it's not required. Blood products, yes. Sometimes questions are asked upon fibrin foam, the storage of the blood products, the type of blood products. So just have a rough, quickly, an overview of the blood products. Certain techniques like biosensors and enzyme immobilizations are or not oftenly asked, but sometimes questions appear, like the technique of adsorption, covalent bonding. They are other types of immobilization techniques. So what are the chemicals used? Like for cross-linking, we use glutaraldehyde, which is a polycross-linker. So that is important. Next up, you have the production of penicillinase, glutamic acid, and citric acid. So basically, these are produced by various bacteria, like Corynebacterium glutamaricum, that is a bacteria for glutamic acid. Aspergillus niger, that is another bacteria. Then you have so many different types of bacteria that are used for production of certain enzymes, certain hormones, certain particular elements. So that is essential. And the last one, but you cannot leave this, that is PCR, polymerase chain reaction, which is a DNA amplification technique. So this amplification technique is very, very important to be studied along with rDNA. So in rDNA, you have vectors, you have DNA ligase, which helps in joining, you have primers. In vectors, you have plasmids, cosmids, bacteriophages. So these and artificial chromosomes. You need to know the difference between plasmids, cosmids, and bacteriophage, and the capacity of how many kilo base pairs they can accommodate. So that is very, very essential when we speak about vectors and PCR. And if I give you a brief overview, okay, हाँ भाई, we are just going to start studying micro and biotech. How should we study? What will be the phases? So phase 1 will be entire vaccine and immunology is to be covered. Phase 2, entire sterilization and staining is to be covered. Phase 3, hybridoma and diagnostic test are to be covered. Phase 4, finally go for rDNA. So immunology and vaccine, sterilization and staining, hybridoma and diagnostic, and rDNA. This is how your sequence would be. So thank you so much for watching today's video. Do like, share, and comment in the comment section, and drop in your queries for free. I'll be more than happy to answer them. Thank you so much for watching the video.